Hello and welcome to this microbiology podcast, Serial Dilutions. Imagine that you're a microbiologist who's been hired to determine the species of bacteria responsible for contaminating the supply of Florida orange juice. To begin, you need to determine the total number of bacteria in the juice. So you take a sample of juice and pour it directly onto a tryptic soy agar plate. After 24 hours of incubation, however, you examine the plate only to discover that a lawn of bacterial growth has formed, making it impossible to isolate individual colonies. Remember that in order to identify specific species, you must be able to isolate single colonies for further testing. So at this point, you know that you somehow need to reduce the number of bacteria in the sample to obtain plates with usable numbers. Most microbiologists prefer to use plates with 30 to 300 colonies. To ensure that the plates produced contain colonies within this range, the original sample must be diluted. Dilution simply means to make something less concentrated. Another way to think about it is that you're watering something down. To demonstrate, assume that you have 4 test tubes, each filled with the same amount of water. To the first test tube, you add 10 drops of blue food coloring, and you watch as the water in the tube turns dark blue. Then you transfer 1 milliliter of the solution in the first test tube to the water in the second tube. The water also turns blue, but it's a noticeably lighter shade than the water in the first tube. You continue this process transferring water from the second tube into the third tube and from the third to the fourth, and you notice that with each transfer, the solution gets progressively lighter in color. This is because you have diluted the concentration of the original solution. This is the same process microbiologists use to dilute the concentration of bacteria in a solution to a level that, when inoculated on an agar plate, will produce usable colonies within the 30 to 300 range. This process is referred to as serial dilution. The most common method of diluting bacterial samples involves a series of 1 to 10 dilutions using 9 milliliter blanks. Each 9 milliliter blank contains 9 milliliters of sterile solution. With each step of the dilution, 1 milliliter of the sample containing bacteria is added to the 9 milliliter blank, diluting the sample by a factor of 10. Because the concept of serial dilutions and the mathematics required to calculate them can get really confusing really quickly, let's take a graphic approach to understanding this process. So let's go back to our example of bacteria in the orange juice. To begin the process, you use a pipette calibrated in milliliter increments and measure out 1 milliliter of bacteria-infected juice, which you add to a test tube containing 9 milliliters of sterile solution. By adding the juice to the dilution blank, you have effectively diluted the sample by a factor of 10, and so we refer to this as a 1 to 10 dilution. The total dilution is written mathematically in exponential form. In this case, the total dilution is 10 to the negative 1 power. When 1 milliliter of the diluted sample is plated on tryptic soy agar and incubated, an overgrowth of bacteria results. Therefore, it's necessary to once again dilute the sample, just like we did when diluting the food coloring in the earlier example. To do so, 1 milliliter of the solution in the first tube is transferred to the solution in the sterile blank in the second tube. Again, because you're adding 1 milliliter of bacteria-laden solution to 9 milliliters of sterile solution, this is a 1 to 10 dilution. However, it's important to remember that this solution had already been diluted once before. To calculate the total dilution, you add the exponents of each individual dilution. In this case, 1 plus 1 is 2, and so the total dilution in exponential form is 10 to the negative 2. This is equivalent to a 1 to 100 dilution. When 1 milliliter of the 1 to 100 dilution is plated, again the growth of bacteria is too extensive to obtain individual colonies. So this process is continued until usable plates are obtained. The resulting dilution scheme is presented here. Take a moment to examine the serial dilution. Be sure you understand how each of the final dilutions were mathematically calculated. Notice that when plated, the 1 to 10,000 dilution produced a plate with 32 total colonies. Using this information, it's possible to determine how many bacteria are in each milliliter of the sample. And the math is simple. The total number of bacteria equals the number of colonies on the plate times the dilution factor. The dilution factor is equal to the reciprocal of the dilution, or 1 divided by the total dilution. So in this case, the dilution factor is 1 divided by 10 to the negative 4, which equals 10,000. So in our example, 32 colonies times a dilution factor of 10,000 equals 320,000 bacteria per milliliter. It's important to keep in mind that this number is only an estimate. When counting colonies, we assume that each one is produced by the replication of a single bacterial cell. In reality, bacterial cells often clump together, forming small chains or clusters that replicate to form a single colony. Therefore, plate counts are more accurately reported as the number of colony-forming units. Given this information, you should now be able to define dilution and understand the process of serial dilution, describe why serial dilutions are so important in microbiology, calculate the total dilution and dilution factors in a serial dilution, and use this information to estimate the total number of bacteria in a contaminated sample.